Biosphere is made from different varieties of living organisms. Now, living organisms, they are also made up of some chemicals, that is elements and compounds. Now, if we perform any analysis of any plant tissue or animal tissue or anything, we obtain a list of elements like carbon, hydrogen, oxygen, and many others. So if we do the analysis of the Earth's crust, that is the non-living matter, we also get the same list. So what's the difference between the two lists? Not much difference. So all the elements present in a sample of Earth's crust, they are also present in a sample of living tissue. But when we closely examine the living matter, we find that carbon and hydrogen are more with respect to the other elements which are present in the Earth's crust. So in this chapter, we'll be studying about all these biomolecules which are related to the living organisms. Topics to be covered in this chapter: how to analyze chemical composition, metabolites, biomacromolecules, proteins, nucleic acids, structure of proteins, bond linking monomers in a polymer, concept of metabolism, living state, enzymes, reactions, then enzyme action, enzyme activity, and various other things related to the enzymes like cofactors. Biomolecules. Now, how to analyze chemical compounds. Now, there are various types of organic compounds which are found in the living organisms. Now, we have to perform a chemical analysis. We can take any living tissue, that is a vegetable or a piece of liver or anything. We grind it into a trichloroacetic acid. Okay, we take any living sample and we grind it with a trichloroacetic acid using a pestle and mortar in a lab. So we obtain a thick slurry. After that, we use a cheesecloth or cotton and we obtain two fractions. One is called as filtrate, which is an acid soluble pool. And the second thing we get is the retentate, which is called as the acid insoluble fraction. Now, when we see how to analyze a living tissue sample, we identify a particular organic compound. We extract the compounds, then we separate them with various techniques till we separate a compound from all compounds. So we isolate, purify the compound, various analytical techniques they are being used in the lab when we separate a compound from a molecular formula. And all the carbon compounds that we get from the living tissues, they are called as biomolecules. So biomolecules, they are present in all the organic matter that is present in the living organisms. Now, if we weigh a small amount of living tissue, any leaf or liver, and we dry it, all the water evaporates, so the remaining material gives dry weight. But if the tissue is fully burnt, all the carbon compounds are oxidized into gaseous form, that is carbon dioxide, water vapors, and are removed. So what we get is the ash. So the ash contains inorganic elements like calcium, magnesium, etc. So when we see various elements which are present in the Earth's crust or in the human body, we find hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, and silicon. Okay, they all are present in various proportions. And when we see the inorganic compounds in any of the non-living matter, inorganic compounds, when we see in the ash, like we have an experiment when we talked about if we burn a leaf or burn a part of a liver and we get the ash and we see the inorganic compounds which are present in them, we find various elements they are present in them. So in the living tissue also we find that sodium, potassium, calcium, magnesium, water and various other compounds, they are present in them. Therefore, this gives us an idea that living tissues with hydrogen, oxygen, chlorine, carbon, they are there in the living tissues. So we can identify the functional groups also from the chemistry point of view, that which functional groups are present in them, like aldehydes, ketones, aromatic compounds. But from a biological point of view, we classify them into amino acids, nucleotide bases, and fatty acids. Amino acids. Amino acids are organic compounds which contain an amine group and an acidic group. You can see here an amine group and an acidic group. Now the acidic group, as it's on the same carbon, that is the alpha carbon, hence they are called as alpha amino acids. They are substituted methanes. There are 4 substituent groups with the 4 valency positions. So these are hydrogen, carbon, and carboxyl group, amino group, and a variable group which is designed as the R group. So based on the nature of the R group, there are many amino acids, but which occur as in proteins, and they are of 20 types only. So the R group in these proteinaceous amino acids, that can be hydrogen, the amino acid is called as glycine. A methyl group, if it is there here, then it is called as alanine. If hydroxymethyl, then it is called as serine. So 3 of the 20 amino acids, they are shown here.